Okay, that's my cue. Hi, everyone. Good morning or afternoon, depending where you're joining us from. Welcome to the Harmful Algal Blooms webinar series. My name's Amy Weckel, and I'm from the Illinois Water Resources Center at the University of Illinois at Urbana-Champaign. I'll be your moderator today. The webinar is hosted by the Algal Bloom Action Team, of which the Illinois Water Resources Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to hosting a webinar series, our team is developing a website of resources, including fact sheets and frequently asked questions on HABS topics. And I encourage each of you to join or visit our website, which I'm going to try to drop in the chat right now. There we go. And there you can explore some of the resources we have to date, including fact sheets on what you should know about HABs and their health-related effects on animals, and prevention and treatment for landowners, as well as a HABs frequently asked questions database and recordings from previous webinars and our virtual HABs research symposiums, which are held annually in January. Before we get started, just a few housekeeping items. We will have a Q&A session after our speaker presents today. Please list your questions for our speaker in the Q&A panel, not in the chat, and we will do our best to address all of them following the presentation. If there is a question already posted to the Q&A panel and you have the same question, be sure to upvote the question so we can get to the most popular questions during the session. Today's presentation is being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you're having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well, and we will be happy to assist you as best we can. And that brings us to our speaker today, who is Ellen Preece, a senior environmental scientist at the California Department of Water Resources. Ellen also serves on the National Harmful Algae Bloom Committee. She has worked on cyanobacteria and cyanotoxins in freshwater and estuarine ecosystems for over 15 years, with a focus on the connection between freshwater and marine ecosystems, with the goal of understanding cyanobacteria dynamics and sustaining the ecological integrity of fresh and estuarine ecosystems while protecting human users. Ellen, I will stop sharing my screen now and you can take it away.
Thank you. Okay, thanks everyone for joining today. This is a talk that I put together with my colleagues Janice Cook from the Central Valley Water Board and Tim Otten and Catherine Cook from Ben Genetics. And I have the talk split into 3 sections. So the first is an introduction to the Sacramento-San Joaquin Delta, which is where my research takes place. And then the second part of the talk is during a drought period, and we're looking at how the seed stock of Microcystis is spread throughout the Delta and how that may source certain areas of the downstream areas of the Delta. And then the third part of the talk is comparing these drought years to this extreme wet year and looking how the Microcystis dynamics are different in a portion of the Delta during that period. So first I want to introduce you all to the Delta. It's this map here on the side. In this brown shaded area is the Delta. The Sacramento River comes in from the north, and that delivers the majority of the freshwater inflow to the Delta. The second major source is the San Joaquin River, which comes in from the south. There are also several other tributaries that flow into it. As the rivers converge, they flow downstream into the San Francisco Bay. So the entire brown shaded region is tidally influenced. So even though it's very freshwater, especially on the eastern edge, it is tidally influenced. There are a number of management activities that occur in the Delta that can also impact the flow of different areas. For example, those two red boxes in the bottom part of the figure, that's the State Water Project and the Central Valley Water Project, and those export large volumes of fresh water to the central and southern parts of California. And when that occurs, it can at times cause reverse flows of the rivers in that region. So there are a lot of different kind of microhabitats across the Delta. You know, we have these areas like the major rivers that have higher flows. We have smaller channels that also can have somewhat higher flows. We have what we call peripheral or dead-end sloughs, and these are have longer resonance times and tend to be shallower. And in our case is where we see the more severe microcystis blooms. And then I'll also be referring to something called the flooded islands. And those are what look like lakes on this figure. They are also, like the rest of the Delta, tidally influenced, but they're more lake-like in nature than some of the other habitats. So microcystis was first detected in 1999 and has been present annually since then at some years, blooms are more severe than others. The potentially toxic cyanobacteria community is generally dominated by microcystis. We actually have picocyanobacteria, which dominates the larger cyanobacteria community. Aphanizonomen and Dolichospermum can also form blooms, but to date, the toxicity has all been linked to microcystis, or I should say most toxicity. There has been some toxicity that's not microcystin linked to the other species. So this is a visual, kind of a conceptual model to walk you through what I was just describing in the last slide. So I'm going to start on this right panel. This is a high-level overview of what the Delta looks like. We have reservoirs upstream that release water downstream. We have riverine nutrient inputs, and then we also have point and non-point nutrient inputs. We have atmospheric deposition that allows nutrients to enter the Delta. We have the water exports that I mentioned. We have stream flow, advection, and tidal forcing that can cause the water and the channels to move in different directions. And then we have these dead-end channels and marinas. So down at the bottom of that is a close-up of A, B, and C. A is the dead-end channels and marinas. These are shallower locations. The photic zone is larger, it reaches the bottom of to those sediments. We get more dense blooms there, which you can see on the right side of panel A. The temperatures tend to be warmer in these locations. You'll notice this 19 degrees Celsius line on all three of the panels. That is the temperature for which microcystis is thought to initiate in the surface waters in the Delta. In the upper left-hand corner, that's the Stockton waterfront, and that is an example of what you see in panel A. You get these dense, oily microcystis scums. In panel B, this is a main channel site. So we have the euphotic zone is smaller. The sunlight doesn't penetrate as far. It's more turbulent. There's higher flows. There's more tidal influence and advection. And panel B is this bottom picture. So we're still seeing microcystis, but instead of those dense colonies, we're seeing these broad lettuce-like flakes of microcystis occur. And then panel C is the flooded islands. Those were the lake-like areas. These are not as deep as the main channel sites, but they're deeper than those static peripheral areas. And we can also get microcystis blooms in those locations. That's shown in that top right-hand picture that says Mildred Island. You can see some flakes of microcystis. There's also some aphanizonomen mixed into that photo. But you can really see how we see the blooms show or present differently depending on the habitat that they're in. So now I want to talk about the weather whiplash that California experiences. First, I want to point out for those of you not in California that there is no normal water year type in California. The state classifies our water year types as wet, above normal, below normal, dry, and critical. And so we're used to having very different water year types annually. It's hard to predict if we're going to have a wet, dry, or in between type year. The drought of 2020 to 2022 was California's driest consecutive 3-year period. And that was followed by 2023, which was one of our wettest years to occur. We had long-lasting snowpack in the Sierra, Klamath, and Cascade mountain ranges. These feed into the Sacramento River and then eventually into the Delta. And I thought this visual from DWR showed really well that difference between the drought and wet years. 2014 and 2015 were also drought years, and then 2021 is one of our study years, and you can see at all of the locations how 2023 is just so much higher natural flow across the state in that year. So now I'm going to get into the first part of the research. This is benthic overwintering cyanobacteria seed stock plays an important role in Delta bloom dynamics, and this was a study conducted during the drought period, 2020 to 2022. So what do we know? We know that some locations are more prone to dense CHABs. The Stockton waterfront and Discovery Bay are two known hotspots, and you'll hear me talking about those locations quite a bit. And then sites in around the central and southern portion of the Delta are also recognized as having more severe blooms. This heat map on the right, which is a chlorophyll heat map, is a good example. It's about a 5-mile transect from Stockton to the Calaveras River. And you can see how the channel is much shallower at the terminus, about 4 meters, and then extends to 12 meters by the end. And you can see that the chlorophyll was much higher in that shallower, kind of longer residence time portion of the channel. This chlorophyll signal was from a microcystis bloom that was occurring in the summer of 2022. And you can see how the chlorophyll spreads out partially because of tides and just natural advection. We also know that microcystis cells deposit in the sediments in late fall as summer blooms senesce. And this figure does a good job of showing that. The microcystis cells, grow vigorously when the surface water temperatures are warm. Then as the days become shorter and there's less light and temperatures cool off, those cells can settle into the sediments where they rest until conditions are good for them to bloom again. And so recruitment into the water column in this figure is 11 to 14 degrees. In the Delta, once again, we don't usually see them in the actual surface waters until about 19 degrees. But it's not clear where microcystis blooms originate in the Delta. Do they develop and grow in multiple areas, or are they mostly originating in those hot spots with the longer residence time? Our hypotheses were that overwintering microcystis seed stock from a few specific locations are sources of CHABs in distant but hydrologically connected locations. By spring, little microcystis biomass will remain in the sediments of the flooded island sites and channels to serve as bloom inoculum. We expected that higher flows during the winter would scour out some of the sediments in that seed stock, plus just natural dying of the cells. And the peripheral sites, Discovery Bay and Stockton waterfront, will carry high seed stock into spring and are the primary sources of microcystis into the Delta, into the southern and central Delta. So high-level overview, we collected sediment so that we could look at microcystis cell abundance. We looked at both total microcystis and the toxic microcystis. We looked at in 1 year when microcystis cells entered the water column. And we did all of this through molecular work. So we quantified microcystis cells in the sediments and water column with molecular techniques, which I'll talk more about. Here's a map of our study sites. And we have these color-coded by the types of habitat. So the yellow sites, these are our two static peripheral areas, the dead ends, where we see the more severe blooms. The green sites, those are the main channels, so where we expect higher flows to occur. The pinkish red sites are the two flooded island locations, so the more lake-like locations. And then the three purple sites, those are outlets. And we're calling those outlets because they are connected to peripheral areas. And then they connect those peripheral areas to main channel locations. For our methods, we collected 5 sediment cores to determine site heterogeneity, and we collected them in November and April for 2 years. So that started in November 2020 and April 2022. The idea is that in November, you'd expect peak seed stock. That's when the microcystis cells should have all settled out of the water column into the sediment. And then April is what you would expect is available for inoculating summertime bloom. And we just have some pictures that we use these sediment cores and pulled them up and then scraped that top portion of the sediment off. The samples were lyophilized or freeze dried. And then we used qPCR to quantify microcystis. We looked at both total microcystis and toxigenic microcystis. And then we used molecular DNA fingerprinting for water and sediment samples so that we can match up different sites to see if there was a relationship between those peripheral areas and the downstream locations. So first I want to show you the heterogeneity that we experienced during our sampling. This is one of the hotspot locations. Please note that the scale is in millions of copies of the genetic material. So even in April, when it looks like there's hardly anything in there, there's actually still quite a bit. But you can really see how there's a wide range in the number of microcystis cells you're going to collect depending on where you drop your core. And you can also see this huge drop off in the number of cells between November and April, which is what we expected. So this is a figure that shows all of the results. I'm only showing on this figure the maximum microcystis that we detected out of those 5 samples. So yellow is November, and this panel on the left is year 1, and panel on the right is our year 2 comparison. So once again, you can kind of see this huge drop off between November and April. The other thing you may notice is that in the second year, there were not nearly as many microcystis cells in November. One hypothesis for that is that we had a major storm event in late October of that year. We got over 5 inches of rain in a single day. And so we think there's a good chance that we kind of missed the peak microcystis in the sediment that November. And then the other thing that's worth pointing out, and you can really see this in the right panel, is how there is more microcystis seed stock in the Stockton waterfront and Discovery Bay, those 2 static peripheral areas, than the other locations. So our summary, we saw a large depletion of microcystis cells at each site over the course of winter, lots of heterogeneity across sites. Seed stock varied by year, possibly due to sampling, possibly due to biomass, or that precipitation event that I mentioned. The highest microcystis in the fall and spring were both at the static peripheral areas, Discovery Bay and Stockton, but Windmill Cove and Outlet and Mildred Island, a flooded island, also had some high microcystis seed stock. So this is a time series of microcystis abundance in the water at each site, and this is from 2021. And what you can see is that there wasn't earlier recruitment at the hotspot locations. We thought there may be, just because the water does warm up more quickly there usually. But what we see is that in July is when we, at most places, see the peak bloom. So that dotted line corresponds when we measured the peak microcystis bloom at each location. And this purple and green dot correspond with Stockton waterfront, Discovery Bay, the 2 hotspots. So have the highest blooms there. The other location that had a high bloom that summer was Buckley Cove, and that is also one of the outlet sites. So those areas with greater seed stock generally had higher surface water microcystis abundance. We didn't see earlier recruitment, but we need more intensive sampling to fully understand the recruitment timing. These are just based on single grab samples. Okay, so I'm going to talk a little bit about some of the DNA work that we did. And so I just wanted to give a high-level overview for people that aren't familiar with that process. So we collected an environmental sample. In our case, we collected water samples. We took DNA from those environmental water samples and then amplified it. So this is using PCR to create a number of different copies of that target DNA fragment. And then it's targeted using a pair of primers. Then it's high throughput sequencing, and this is simultaneously sequencing these millions of DNA fragments. Then it's a bioinformatic processing step, and this applies computational approaches. So we were comparing our microcystis genotypes to microcystis genotypes that occur in libraries, genetic libraries. And then you can identify your species. In our case, we were identifying the different microcystis genotypes. We know that there's multiple microcystis genotypes in the Delta, but we didn't know how many. And so that was what we were trying to do here. And then based on that genotype identification, we can develop these molecular fingerprints for each of our sites and then try to match them up to other sites to see if there's a relationship between these hotspot locations and other areas where we also observe microcystis blooms. So, based on that DNA work, we developed a dendrogram and heat map. And I'm just gonna spend a little time walking you through this. So we found 36 microcystis genotypes in the 2021 Delta water samples. The color change from blue to red is indicating increasing abundance. So the warmer or redder the color, the greater the abundance. And we had 6 clades. So a clade is a group of biological taxa. So they're similar. And so the first clade primarily consisted, this top clade here, of main channel sites, outlets, and flooded islands in June, July, and August. It indicates that these areas had high exchange of microcystis during the summer. Clade 2 is only Discovery Bay. That's the only site that matched up with it. And it's a unique strain composition in early July. And just real quickly, for color coding, the blue on the far right here, those are the main channel sites. The purple are the outlet sites. The greenish color are the static peripheral or hotspot sites, and the red is the flooded island sites. So moving on to clade 3, it was very small and consisted of rare strains from Stockton and then also Buckley Cove. Buckley Cove is about 5 or 6 miles downstream of Stockton, and the water does flow from Stockton down to Buckley with tidal exchange. So there may be some transport between Stockton and Buckley Cove. Clade 4, we really see a shift here in late summer for Clade 4 for the outlets, Windmill Cove and Buckley Cove, and the main channel sites in Old River. Clade 5, it really stands out that it's a large variable group, but it's mostly those static peripheral or hotspot locations. Strains are present at lower abundances at a couple of other sites, but primarily Discovery Bay and Stockton. We don't think, based on the known hydrology of the area, that these two locations are exchanging cells. Instead, we hypothesize that environmental conditions are similar at the two hotspot locations, and that's why the cells may match up there. Lastly, clade 6 is kind of an amalgamation of disparate sites and strains, and the only unifier is that they were all collected in late summer. You can see it's all August and September dates. So perhaps late summer conditions favor the strains or genotypes in clade 6. I'm not going to show you this for time, but we have this same map for the toxigenic microcystis, or the same dendrogram for toxigenic microcystis as well. So next, this is a microbial network occurrence analysis. And so we're only looking, we put all of our samples into this, and we're only looking at the relationships that exhibited significant and strongly positive Spearman rank correlation. But there's a few things that I want to point out. So it's the same color coding as the dendrogram. But first, we see this connection between the outlets of Windmill Cove and Buckley to the main channel site of Old River and to the flooded island sites. Second, we see a relationship between Stockton and the San Joaquin River deep watership channel. This is just downstream of Stockton. And then the third relationship that we see is that Discovery Bay, so that static peripheral area, is connected to its outlet, Indian Slough. We also have the same figure for the toxigenic microcystis, but I don't have time to share that today. So molecular summary, based on the two figures I just showed you, the two figures that I didn't show you, and an NMDS analysis, we identified three connections. The first is what we're calling the west side connection. It appears that there's a relationship between the microcystis cells in Discovery Bay, along Old River, over to the two flooded island sites. Then we have what we call a north side connection. This is that cells over in the Buckley Cove and Windmill, so these two outlet sites are then connected to the two flooded islands and Old River. And then our final one, kind of building on what we call the north side connection, and this is connecting the Stockton area to Windmill Cove and Buckley Cove, so number 4 and 3 on this map, and then also ending up in Frank's Tract and Mildred Island. So conclusions. This was the first study to investigate overwintering microcystis cells in the Delta. Microcystis at some locations are likely seeded from other upstream locations in the Delta. It's likely that there's multiple locations that maintain microcystis seed stock into the spring that could initiate blooms. So we had expected that Discovery Bay and Stockton they did carry the most seed stock, but we were surprised that some of the outlets and that Mildred Island also had a decent amount of seed stock going into the spring. It's not possible to conclude what % of overwintering cells died versus those that were transported to other Delta locations in the winter. So next steps were already underway for a more intensive seed stock studying. We have a larger number of sites. We're doing the major surveys in November and April again, but we're also conducting monthly sediment and water sampling at a subset of sites, including the Stockton area. And that started in November 2023, so we'll get to see if the wet year had any impact. We're assuming it will, because microcystis bloom for one of the first times ever in Stockton that year. And I'll talk about that a little more in the next section of my talk. We also have teamed up with UC Merced to do some laboratory-based studies. We want to better understand seed stock recruitment when environmental factors are triggering those cells to emerge from the sediments. And then we're also going to test some potential mitigation measures to look at potential treatment of the seed stock to help prevent blooms in the future. I'm going to now move on to the next part. And so the title of this part of the talk is Use of Multiple Sampling Modalities for Cyanobacteria and Cyanotoxin Monitoring in the Sacramento-San Joaquin Delta under different hydrologic regimes. And this covers the 3 drought years and that extreme wet year. So previous work has shown that droughts promote more extreme CHAB events in the Delta, yet even during really wet years, CHABs are not completely eliminated from the Delta. And since we had this significant meteorological contrast between the drought years of 2020 to 2022 and 2023, we had a unique opportunity to compare water quality and cyanotoxins across a range of conditions. And I think this picture, at least visually, summarizes really well what we saw at the Stockton waterfront. During that critical drought year of 2022, we have this oily microcystis scum. That same view in 2023, when we did not observe any microcystis at this location, you can see that macrophytes are utilizing that same space. So the objectives of this work were to compare water quality conditions between drought years and a very wet year, compare toxic cyanobacteria detection methodologies by comparing integrative samplers. So we used solid phase absorption toxin trackers, or SPAT samplers, and we also used invasive Asian clams bivalves, along with water grab samples and some more molecular work. We also examined the relationship between toxin-producing cyanobacteria and cyanotoxins relative to environmental conditions. So we looked at nutrients, temperature, and hydrology. So for this map, we have 3 primary sampling sites. You may recognize some of the names from my last talk: Old River here, Middle River, and then the Turner Cut location. And then we were also able to include Stockton waterfront in this study, but only were able to look at toxins in water samples at that location. So for our field work, we collected water grab samples between July and October of all 4 years. We looked at nutrients, toxins, and then molecular work on those samples. We collected environmental parameters in the water with the BIOSI data. We also used invasive clams at each of those sites over the 4-year period. And then we were only able to deploy SPAT samplers in 2023. In addition to the fieldwork, we also acquired data from fixed stations. There are fixed stations that occur at those 3 primary sampling sites, and they collect a variety of real-time data, including temperature and flow data. And then we are also able to utilize cyanobacteria harmful algal bloom response monitoring toxin data from the Central Valley Water Board. I'm going to jump into the results. So first, going to share this principal component analysis that we did. So this was looking at all of the different factors that we had data for 4 years at those 3 primary sites. And the years are color-coded on here. And what you can see is that 2023 separates out from the other 3 years. 2020 and 2021 are more associated with the right side of the plot, where higher nutrients, higher microcystin, and the watering clams. It's also interesting to note that 2022 separates out. So recall that 2022 was also a critically dry year, along with 2021 and 2020. But it separates out from 2020 and 2021, and we observed that in numerous instances in our data, and I'll point that out as we go along, but it's interesting that that very dry year was different than the other 2 very dry years. So first, we looked at temperature. So this is the time series of the 4 water years between 2020 and 2023. Those gray shaded areas are July to October. And that dotted line is that 19 degrees threshold for when we expect to see microcystis. So using a linear mixed model, we did not see any statistical difference in temperatures across years. And just visually looking at this plot, that makes sense. We also looked at when that 19 degrees threshold initiated, and it was in April of all 4 years. So even 2023 was not cooler at our study sites compared to those drought years. Next, we looked at the flow data from those stations. Once again, shaded areas are that July through October period. And what I wanted you to see here is look at that increase in springtime flows in 2023. That's a huge difference compared to the other years. Old River and Middle River, by summertime, those flows had returned back to kind of a baseline condition, likely in part at least due to the water exports that pull water from those locations. Turner Cut is a little less influenced by those water exports pulling the water. And a close-up of that, so this is a different scale of that same Turner Cut plot. You can see that flows at the Turner Cut site during the summer months were about 500 CFS higher than they had been during those drought years. And this is likely related to the San Joaquin River flows because the San Joaquin River is just adjacent to the Turner Cut site. Next, I wanted to talk about nutrients. There's a lot going on in this plot, but I just wanted everyone to see that we have pretty high nutrients in the Delta. The nutrients were non-limiting at all sites and years, but total phosphorus was significantly lower in 2023 relative to 2020 and 2021. 2022 total phosphorus was also significantly lower than 2020 and 2021. So that's an example of, even though 2022 is this extreme drought year, it's separating out from 2020 and 2021. And then total nitrogen was also significantly lower in 2023 relative to 2020 and 2021, but not 2022. Moving on to microcystin from our water grab samples. Now we can bring Stockton into some of our figures. And you can see that the Stockton waterfront, which is that blue dot, can have really high concentrations of microcystin. In 2020, exceeded 1,000 micrograms per liter. And although it wasn't quite as high as in 2021 and 2022, you can still see that it exceeded the California danger threshold of 20 micrograms per liter on numerous occasions. The Old River, Middle River, and Turner Cut sites have much lower concentrations of microcystin than that hotspot Stockton waterfront site, always below the California warning threshold of 6 micrograms per liter. But what's interesting to note is that in 2022, those 3 sites, Old River, Middle River, and Turner Cut, had no detections of microcystin. And in 2023, no water grab samples at any of the 4 sites were positive for microcystin. This is a figure showing the Asian clams. So we used enzyme-linked immunosorbent assays to measure microcystin in Asian clams. We also used LC-MS on a subset of our samples to confirm our ELISA findings. And this shows the 4 years of microcystin in those Asian clams. And what you can see is that 2020 did have significantly higher microcystin in clams than the other 3 years. The other 3 years didn't separate out, but it's interesting that in 2022 and 2023, we did still detect microcystin in clams, even though we did not detect the toxin in the water grab samples. In 2023, 6 of the 11 spat samplers were also positive for microcystin at these sites. So that confirms that microcystin was present during the wet year. We weren't picking it up with those water grab samples. This is some molecular work that we were able to do on our samples. We only are able to show you 2021 to 2023 in this site, or in this plot. So we're missing 2020 when we had the highest toxin concentrations in the clams. But panel A on the top is an estimate of total cyanobacteria. Those are those black squares, and microcystin producing cyanobacteria, which are the red circles. And we saw the most cyanobacteria in 2021, 2022, and less in 2023. But it was still present, and that MCYE gene was present in our 2023 samples. On the bottom, that's the relative abundance of cyanobacteria genera from all samples with the detectable MCYE genes. And you can see, so that red kind of striped area, that's microcystis. Microcystis was most present in 2021, but you can see that we did detect a little bit with these molecular methods in 2022 and 2023. And those blue bars are the picocyanobacteria. So I mentioned that at the beginning of the talk, that dominated the cyanobacteria community. It's also worth noting that all MCYE sequences were closely related to microcystis. So This builds off past work by Tim Otten that shows that Microcystis appears to be the sole producer of microcystin in the Delta. And then I'm not showing Stockton on this figure because our data set was limited, but we do have 4 samples from Stockton in 2023, and there was no MCYE in 2023. So Stockton, there's no evidence of microcystis in any of our samples in Stockton in 2023, which is very unusual. So to wrap up what we found, we expected microcystin concentrations in Asian clams would be lower in 2023 relative to the drought years, but it was present in all clam samples collected from 2023. The microcystin concentrations were highest in 2020, followed by 21 and 23. And interestingly, 2022, which you'll remember, is that third drought year had the lowest concentrations of microcystin. And that year also we saw no microcystin in the water column. Spat samplers confirmed microcystin presence in 2023. Molecular samples confirmed the MCYE genes were present at 3 of those 4 sites. And if we had only relied on water grab samples, we would have missed the toxin presence in 2022 and 2023. So comparison between years, water quality conditions were different between the drought years and the wet year. The significant differences that we found in our data set was that spring flows were higher at all sites. Summer flows at Turner Cut were higher. Nutrients were lower, but there were still plenty of nutrients in the system, and microcystins were lower. No microcystin or toxigenic microcystis cells were at the Stockton waterfront. And this is, like I keep mentioning, really unusual. This is a hotspot site known for annual microcystis events. In 2023, the presence of toxigenic microcystis cells combined with microcystin in clams at all sites suggests that even in these record-breaking wet years, conditions in at least some locations of the Delta remain sufficient for microcystin production. What's next? We don't have a direct study like we do for the last talk I gave, but we do expect that temperatures will increase and this under future climate scenarios, and this will exacerbate blooms. And there's also a pressing need to implement a monitoring program in the Delta. We want to make sure we capture these toxic CHAB events at appropriate temporal and spatial scales. And our work shows the importance of considering multiple sampling modalities while doing that. I just want to thank everyone that helped with field sampling and data analysis for this project, and then funding from Proposition 1, California Department of Fish and Wildlife for helping fund the second portion, and Delta RMP and the State Water Board and Central Valley Water Board for helping to fund the first portion of my talk. And that's all.
Great, thanks, Ellen. Okay, if you want to stop sharing, I can share my screen again. And so yeah, I'd like to thank everyone for participating in the Algal Bloom Action Team's webinar series. A special thanks to Ellen for sharing her research with us. And also a thank you to Ann Nardi and her team at the North Central Region Water Network for hosting the Zoom webinar and coordinating the technical aspects of the webinar. We couldn't do it without their team. As we move into the Q&A session of the webinar, I'd like to remind you to check out the Algal Bloom Action Team's website. There you'll be able to connect with the team and subscribe to our events, such as our webinar series, which you're in right now, and it's held bi-monthly with the next one scheduled for June 5th. We'll now move into the Q&A session of the webinar, and we'll draw the initial questions and comments from those submitted in the Q&A panel during the presentation. So the first question I have, let's see, we'll do the most upvotes. What 3 main water types are the depths on the graph in centimeters or inches?
The depths. Oh, those were in meters. The depths on the figure were, I think you're talking about the heat map that I showed. So the static peripheral areas, like the Stockton waterfront's about 4 meters. Um, the flooded islands are, depending where you go, 6 to 10 meters. And then those main channel sites are 12 meters.
Okay. Hopefully that answers your question, Bryce. Ed says, for benthic microcystis blooms during drought conditions, do you think there is sufficient internal recycling of nutrients, legacy phosphorus, to sustain such growth during such dry periods?
We have started to look into some phosphorus in the sediments, and the pore water is within the sediments, and it's definitely higher than the water column. And the UC Merced group is also going to investigate the sediment composition more and try to better understand what that source of nutrient loading is. But the Delta is just really well recognized for having a lot of external phosphorus and nitrogen loading during both dry and wet periods. So it's probably a combination, but the system just has a lot of nutrients and stormwater. There's a lot of stormwater that also delivers nutrients into it.
For the molecular analysis where you showed the network of connections among each sampling location, could you briefly mention the methodological approach you used to get these individual connections?
Yes, so we did a network analysis and used Spearman's ranks correlation and looked at a positive relationship of R greater than 0.75. Anything that was positive of greater than 0.75 was included in the network analysis.
And what types of nutrients were you looking at, and how did you analyze for them?
Yeah, so that was the box plots that I showed. I maybe went through it a little too quickly, but we looked at total phosphorus, phosphate, total nitrogen, nitrate, and ammonia. And we just used standard colorimetric nutrient analysis at Ben Genetics, ran all of the samples for that.
Where did you have well, there you go. You answered, I think you just answered that. Where did you have the samples analyzed, the differentiated species?
Tim Otten's lab, Ben Genetics, analyzed all of the samples, but that DNA work, he sent the filtered samples, DNA samples, to Oregon State University, and they did the DNA molecular processing for us.
Okay, and Hannah is curious about the life cycle of the clams. In the case of 2023, where no microcystin was found in water samples, but it was found in the clams, could that toxin be remaining in the clam system from years previous?
We don't think so. The clam study, so the project that I presented built off a larger microcystin clam study, where we looked at microcystin clams at 10 locations in the Delta for a 2-year period every month and even more in the summer. And we calculated a deparation rate based on our findings. And we think that microcystin depaраtes out of the clams after about 10 days. So we don't think it was the previous year's microcystin.
Okay, if microcystin is not sufficiently concentrated in some years to detect in grabs, for example, clams or spat only, oh, that just jumped. Is it still a human health risk or just an academic curiosity?
So the larger clam study that I just mentioned and that this project utilized that data and built off of, the purpose of that study was to see if the benthic food web was contaminated with microcystin. We have a number of threatened and endangered fish species that feed in the Delta, and we wanted to see if it was a potential stressor to the food web. We don't have any thresholds that we can compare like we do for human health, but we can say if it's present, then it's a potential stressor. And there's a lot of interest in protecting these fish species. So I would say in that case, it was about food web and ecosystem effects. And then it's also worth mentioning that those clams that are an invasive species are very small, but there are certain subsistence fisher groups that collect and eat them. And so there's also a number of other shellfish and organisms in the delta that potentially accumulate the toxins and may be a source of toxins that, especially to subsistence consumers that are eating those. So I would say it's more than just an academic curiosity.
Great, thank you. Why do you think there were no microcystis at Stockton in 2023?
Well, I don't know exactly. That's the big question. Just some observations that we've brainstormed about is that there was a diatom bloom instead. And it did seem like the air temperature was cooler at that site in June than the previous year. We haven't analyzed that site's comparing the temperature data in the water, but I was thinking maybe the diatoms kind of got a hold of that area and were able to then outcompete the microcystis. But I don't really know. It'll be really interesting to see if there's any of the microcystis in the seed stock when we analyze our 2023 samples. Maybe that seed stock got flushed out during the 2023 precipitation events. So those are some ideas, but that's like the million-dollar question. We don't know exactly.
For your grab samples, how many duplicates did you collect per site? And did you do multiple grab samples of each site over time or season?
I don't think I explained that well. We only had single grab samples. Although we always collected at least one duplicate at one of the sites throughout just for quality control. And we are collecting those samples twice per month in July, August, September, and October.
And I think we've got one final question. When you and your team identify dangerously high levels of toxins, are the results transferred or shared with the public health and safety departments? And are the sites flagged?
So that data that I shared with you at Stockton that were the dangerously high toxins, that was data from the Central Valley Water Board, and it was specifically response sampling. So yes, they communicated that concern to appropriate people.
Great. Okay. Well, thank you again, Ellen. I don't see any further questions. So yeah, thank you for your time, and thanks everybody for participating in the Harmful Algal Bloom webinar series. We'll see you again back in June, I hope.
Yeah, feel free to email me with further questions as well. Thank you.
Thank you.